Hello everybody, you are welcome. Today we will start revision of paper 4, classified chapter 1, energy and respiration. Question number 1 from page 41, May June 24, question number 1. Let's start. Oxygen is needed for aerobic respiration in cells. In eukaryotic cells, the mitochondrion is the organelle used for aerobic respiration. Letter A, figure 1.1 shows a transmission electron micrograph of mitochondrion. A, B, C, and D for letters. Okay, here table 1.1 lists the 4 stages of aerobic respiration. With reference to figure 1.1, complete the table 1.1 using the letters A to D to show where each stage occurs, precise location of each stage. Each letter may be used once, more than once, or not at all. Stage of respiration, glycolysis. As we know from our study, glycolysis takes place inside cytoplasm. So we will choose letter B. Okay? Glycolysis where glucose is split into 2 pyruvate. This process takes place inside cytoplasm. Then what about link reaction and Krebs cycle? Link reaction and Krebs cycle both happen in the matrix of mitochondria, so we will choose letter D and D, okay, for both. Last stage of aerobic respiration is called oxidative phosphorylation. It needs respiratory complexes, okay, several enzymes, and ATP synthase enzyme. All of them lie on the inner membrane of mitochondria, I mean crest, okay, so we will choose the letter C. Okay, in anaerobic conditions, no ATP can be synthesized by oxidative phosphorylation because the process stops. Explain why ATP synthesis by oxidative phosphorylation stops in anaerobic conditions. Here, as we know, oxygen is not needed in glycolysis, in link reaction, and Krebs cycle. Only it is needed in the last stage of aerobic respiration, I mean oxidative phosphorylation. When oxygen is absent, or when oxygen is not enough, not sufficient, what would happen? Oxygen acts as a final electron acceptor in the electron transport chain. Here part of mitochondrion shows outer and inner membrane, and between both membranes there is chamber or compartment called intermembrane space. Okay, in the inner membrane of mitochondria, a lot of enzymes or carriers, okay, respiratory enzymes or electron carriers, plus ATP synthase, all of this protein found and embedded within the inner membrane. Here oxygen, look at oxygen, have O2, one oxygen atom is waiting for what? Waiting for electrons, but these electrons must be lowered in energy. When NADH and FADH2, okay, released their protons, lose their protons and their electrons, okay, electrons with high energy must be lowered to be low energized as the last stage. How come? By passing these electrons through electron carriers within the membrane. During this process, what would happen? Step by step, electrons release excess energy. Okay, this energy is used to pump protons, okay, from FADH and FADH2 and NADH is used for pumping protons towards the intermembrane space. Electrons finally is lowered in energy. Now they are ready to combine with half oxygen and 2 protons to form water in process called chemiosmotic phosphorylation. Okay, look please. Here, explain why ATP synthesis by oxidative phosphorylation stops in anaerobic conditions. All this reaction stops. Why? In anaerobic conditions, the absence of oxygen stops the electron transport chain. Why? Because NADH and FADH2 cannot be oxidized to form NAD plus or FAD. cannot be oxidized. So without NAD, the Krebs cycle and link reaction cannot be continued. As we know, in Krebs cycle, 6 NADH are formed. During link reaction, 2 NADH are formed. Okay, so totally 8 NADH not formed at the time of absence of oxygen. Okay, now ATP production by oxidative phosphorylation stops also, leaving only substrate level phosphorylation that was happened in cytoplasm before pyruvate try to enter the outer membrane of mitochondria. Okay, up to 60% of ATP that is produced in cancer cells comes from lactate fermentation of glucose, even though oxygen is present. Okay, cancer cells preferred lactate fermentation of glucose even though oxygen is present. Scientists are developing cancer treatments to inhibit the enzyme that catalyzes the last step of lactate fermentation. Explain how the inhibition of this enzyme reduces the production of ATP in cancer cells. From our study, in anaerobic respiration, okay, in this stage log, in this process, lactic acid fermentation, glucose is splitting into pyruvate, 2 pyruvate molecules in cytoplasm. Okay, here pyruvate need to be reduced by hydrogen carried by NADH. Okay, NADH, 2 NADH. Pyruvate uses hydrogen carried by coenzyme reduced NAD. Okay, this taken or split from NADH, this hydrogen split or released from 2 NADH by enzyme called lactate dehydrogenase. Scientists developing cancer treatment, okay, try to inhibit this treatment, okay, try to inhibit this enzyme in last step. So inhibition takes place for lactate dehydrogenase. What would happen? What's the mechanism of this new treatment? Look, here the inhibition of lactate dehydrogenase enzyme prevents lactate production of sugar, okay, and NAD plus regeneration. NAD plus as coenzyme is limited in cells, is limited in limited amount in cells. So here, due to inhibition of lactate dehydrogenase, hydrogen is not removed from NADH, and NADH is not oxidized to form NAD plus. Now, without NAD plus, glycolysis stops. The reaction from beginning stops without NAD, because we search for certain carrier of hydrogen split of glucose, where no carrier, no proton carrier, so glycolysis stops. Now this reduces ATP production of sugar, because the glycolysis is the main source of ATP in cancer cells, 60%, and here 60%. Okay, from beginning 60% of ATP that is produced in cancer depend on lactate fermentation. So this lead to decrease the chance of dividing cells of cancer or the size of tumor. Okay, question number 2 from paper 4, variant 2, May June 24, question number 6. Okay, here figure 6.1 is diagram of part of mitochondrion. Okay, here outside cytoplasm, intermembrane space, outer membrane, inner membrane, and in between intermembrane space. Here matrix of mitochondria. On figure 6.1, use arrows to show the route taken by oxygen molecule from cytoplasm into the mitochondrial matrix. Oxygen lie here. Okay, how oxygen reach the matrix? What about its route? As we know, oxygen is a gas. Oxygen is non-polar molecule with no charge, so easily pass through the phospholipid bilayer of both outer and inner membrane of mitochondria. So passively diffuses across the membrane. Okay, no need for what? For carriers or transport to protein or channel. The same also with carbon dioxide here, produced due to aerobic respiration, also passes passively to outside. Okay, with the same route. Why? Because CO2 is a non-polar compound. Okay, it passes easily through hydrophobic phospholipid bilayer of both membranes. Now, letter B, identify protein D. Where is D? Here, this protein. And the group of proteins represented by A, B, and C. And describe their rules in oxidative phosphorylation. Wow, 7 marks. Okay, let's break down these marks together step by step. Look please at this presentation. Okay, here is. Okay, here in this presentation, look at this diagram. Cytoplasm, inner membrane, outer membrane. What about these proteins? D with channel specified for protons to pass from intermembrane space into matrix, return back. Look at these protons. Okay, here channel within the ATP synthase. So protein D is ATP synthase enzyme. Okay, what about other proteins A, B, and C? All proteins with purple color embedded in the inner membrane space act as proton carrier, okay, or electron carrier, where NADH, okay, releases its electrons and its protons to change into oxidized form of NAD+. FADH2 releases electrons also and change into oxidized form FAD, okay, within this electron carriers. Electron released from both NADH and FADH2 carried by electron carrier within the membrane to be lowered in energy. Okay, the purpose of electrons transfer is to lower their energy. They here in the first step, they are very high in energy. Okay, the energy present in electrons not captured by cells, not useful, not harnessed or harvested by cells, so it must be lowered in energy. How come? By passing through electron carriers within the membrane. Okay, now look at this video for ATP synthase demonstration. But before starting this video, I need to remind you here certain mistake. This not cytoplasm, this matrix of mitochondria. Okay, here matrix of mitochondria, matrix. Okay, matrix. Here the inner membrane of mitochondria. Here intermembrane space, okay, intermembrane space. What about here this outer membrane, for example? Okay, outer membrane. All letters mentioned by narrator in this video not needed. Okay, just I need gamma here, gamma axial, okay, or gamma axial, okay. Now let's start this video after erase all ink. Ready? Okay.
A concentration gradient of protons across a membrane drives the synthesis of ATP by a large complex in the membrane called ATP synthase. ATP synthase, also called the F1-FO ATP synthase, has two main parts: F1, which protrudes into the cytoplasm, and FO, which sites in the membrane. FO consists of 12 identical subunits designated C. A central stalk called gamma subunit extends from FO through F1. Surrounding the gamma subunit in F1 are 3 pairs of alpha and beta subunit, which serves as sites of ATP synthesis. Other subunits designated A and B connect the F1 knob to the membrane. The proton gradient across the membrane drives FO to rotate. Protons enter through a channel in subunit A and moves into the C subunits, causing FO to rotate in 120 degrees steps around the axial. Protons are released from the C subunits into the cytoplasm. Note that the FO rotates, the axial also rotates, but F1 does not. F1 is held in place by its connection to the membrane through A and B subunits. However, the axial turned inside F1 causes F1 to change conformation, catalyzing the formation of ATP from ADP and inorganic phosphate. An ATP molecule is produced for every 3 protons that crosses the membrane through the ATP synthase channel. This function, in which a proton potential powers ATP production, is highly conserved. ATP synthetase complexes with the same general structure can be found in the cell membranes of bacteria, in the inner membranes of mitochondria, and in the thylakoid membranes of chloroplasts.
Okay, thanks. Look please, here letter D, ATP synthase. A, B and C are electron carriers, carry electrons with high energy to be lowered. Okay, in the last step, look please, description of their rules. ATP synthase uses the proton gradient established across the membrane, inner membrane, from intermembrane space towards the matrix through the inner membrane to synthesize ATP from ADP in oxidative phosphorylation. Okay, here the flow of 3 protons through ATP synthase provides the energy required for this reaction. Okay, we need to bind ADP plus BI by energy from where? From flow of protons depending on gradient proton potential across the inner membrane. Here electron carriers A, B and C. A, B and C involved in transferring electrons are needed for carrying electrons through the electron transport chain. Here, this electron transfer facilitates and provides the energy needed for the active transport of protons. H from the matrix, here proton pumping, H proton from the matrix into the intermembrane space, creating proton gradient. How these protons accumulate in high concentration in intermembrane space? By this, look please, by here bumping the electron, release energy all the time. Okay, release energy because it moves from high state to ground state to lower state. It releases a lot of energy. This energy is harvested and used to pump protons, okay, from matrix, okay, towards the intermembrane space. Okay, need energy. Now again, look please, this called what? This process is called chemiosmotic or chemiosmosis. Why? Because electrons with low energy combine, two electrons here, okay, 2 electrons with low energy and have oxygen as the final electron acceptor plus 2 protons combine together to form water, H2O. This is called chemiosmosis. Okay, now letter C. The matrix of mitochondrion contains small circular DNA. In the cells of humans, the mitochondria have circular DNA that contains 37 genes. Suggest functions for these mitochondrial genes. Okay, with 3 marks. Look at the structure of mitochondrion. Several copies of DNA, circular double helix, double-stranded DNA. Several copies here and here and here. Several copies found in mitochondrial matrix. Each DNA molecule carry the same 37 genes. Okay, what's the importance? What's the function of this mitochondrial genes? Okay, genes carried by DNA are very, very important. They carry codes for production of several proteins needed for aerobic respiration. Like what? Like genes for ATP synthase as enzyme. all enzymes are protein. All protein need code on DNA. Okay, so ATP synthase is expression of certain of these genes. What about ribosomes? Ribosomes also mitochondrial ribosomes with 70S. It is combination between what? Between rRNA and proteins. How these machines for translation of messages are formed inside mitochondria? By genes of circular DNA. During protein synthesis, okay, we need something called tRNA. tRNA transfer RNA that carry amino acids, okay, to ribosomes, and ribosomes starts to assemble this amino acids according to sequence of codons on mRNA in protein synthesis. Okay, look please step by step. The genes, some of the genes code for essential proteins involved in oxidative phosphorylation, I mean ATP synthase enzyme and electron transport chain proteins. Okay, include membrane proteins like transport or carrier protein like porin in the outer membrane of mitochondria, porin, where pyruvate passes through. Here rRNA genes are used for mitochondrial ribosomes formation. Ribosomes are formed by combining rRNA molecules with proteins. So genes on DNA are needed to be transcribed into rRNA. Okay, plus tRNA molecules are required for mitochondrial protein synthesis, where they carry amino acids to reach ribosomes to be assembled into polypeptide chains. May June 24, question 8. Letter A, figure 8.1 is diagram of mitochondrion, where outer membrane and enfolding inner membrane outlines the rules played by the mitochondrial membranes in respiration. What the rules of mitochondrial membranes in respiration? Okay, with 3 marks. Okay, look please. Here, outer membrane carry a lot of pores, gated or surrounded by porins. Porins, this type of protein, okay, found on the outer membrane of mitochondria. What else? Inner membrane, outer membrane firstly. Outer membrane acts as barrier, separating the contents of mitochondrion from the cytoplasm. Barrier, okay. Here type of compartmentalization. Membrane in general in bio act as or provide compartmentalization for each organelle to be separate from other organelles or reactions that takes place in cytoplasm, for example. So outer membrane acts as barrier, separating the contents of mitochondrion from the cytoplasm. Here contains protein channels, porins, that allow small molecules such as pyruvate to enter to reach link reactions and then Krebs cycle. Inner membrane, as we saw before, impermeable to most ions. Why? To maintain or keep electrochemical gradient and the pH inside matrix, where a lot of enzymes lie in matrix. Enzymes for link reactions, enzymes for Krebs cycle reactions, all of these enzymes need certain and proper pH. So inner membrane must be impermeable to most ions, maintaining electrochemical gradient required for oxidative phosphorylation. Here, site of electron transport chain, we found several electron transport proteins and electron carriers, okay, mobile carriers within membrane, where electrons from NADH and FADH2 are transferred through complexes. Also creates proton gradient. This functions of both membranes creates high concentration of protons inside intermembrane space, so create proton gradient high concentration of protons inside intermembrane space and low within matrix, that create proton gradient. okay, and create energy by pumping or passing protons within the channel attached with ATP synthase. This create energy, okay, energy needed or enough to bind Bi with ADP to form ATP in process chemoosmotic phosphorylation. Okay, contains ATP synthase also is a protein which is synthesize ATP as proton flow back into the matrix through it. Here intermembrane space accumulates protons during the ETC, okay, electron transport chain, establishing the proton gradient. Okay, now letter B with 2 marks. The shapes and the numbers of mitochondria are continually changing due to fission. Fission occurs when one mitochondrion splits into or to form 2 mitochondria. Suggest reasons why mitochondria carry out fission. As we know, here for mitochondrial DNA and the mitochondria itself, it starts to split into two parts, 2 mitochondria. This type of division, it behave independently on nuclear division, on mitosis, for example, independently. Look, please. Here, to increase mitochondrial, why mitochondria carry out fission? To increase number of mitochondria, of sure, increase in number, to meet higher energy demands, as during cell growth or division. Here, certain facts. When we exercise daily, what would happen to our muscles? They possess more and more mitochondria. This exercise encourage the binary fission of mitochondria to reach maximum number. Why? To provide the required energy by your muscles. Okay. Now, to isolate and remove damaged parts, ensuring the mitochondria remain functional, where mitochondria by their activity in cells, time after time, okay, after doing this several times, a lot of reactions takes place, it become elderly or old, so it must be replaced by another new one. From where? From binary fission of mitochondria itself. Okay, to isolate and remove damaged parts, ensuring mitochondria remain functional. Look also, to allow redistribution of mitochondria to areas of the cell requiring more ATP. This binary fission provides a lot or generation of new mitochondria. Okay, this redistributed or move along the cell to reach certain part of the cell where energy is in more demand. Look please at this figure. Here a cell of our kidney tubule. Look at this part of cell where mitochondria lie a lot here. Why? Because this part of cell needed to release energy for pumping certain molecules like sodium ions, for example. Okay, we will study this in later lesson, inshallah. Look please, guanosine triphosphate, GTP, is a molecule that is used as a source of energy in some reactions instead of ATP. Guanosine is composed of purine, similar to adenine and ribose. Suggests why GTP can be a suitable source of energy in some reactions. Here, comparison between ATP and GTP. ATP, adenosine triphosphate. What about GTP? Guanosine. What is meant by guanosine? A base called guanine attached with ribose. Here, ribose sugar. Okay, guanine. Okay, guanine. Okay, guanine plus ribose called guanosine, and 3 phosphate groups. It is similar in structure, but only we replace adenine with guanine. Okay, now look. Here, certain energy coin, cellular energy coin. Instead of ATP, we use sometimes GTP. It easily hydrolyzed to form GDP, guanosine diphosphate, and this allows certain molecules to pass across the membrane of our cells. Okay, GTP. It easily hydrolyzed by adding water by certain enzyme. Certain enzyme is needed. Look please, GTP like ATP has high energy bond. Okay, that can be hydrolyzed to release energy. During release of this energy is needed to change conformational change, 3D change of certain protein across the membrane to allow certain molecules to pass in or out of the cell. G proteins binds to GTP rather than to ATP. Certain protein or membrane protein is called G protein. Sometimes we use this G protein in certain examples, okay, to allow certain molecules to pass through, okay, or make change as cellular communication. It is not important to pass something across the membrane, but may trigger certain reaction within the cytoplasm. It's okay. Letter C. Old or damaged mitochondria reduce the ability of a cell carry out aerobic respiration and produce the ATP needed for metabolic processes of the cell. Suggest what occurs to this mitochondria to allow the cell to maintain the same overall rate of respiration and ATP production. Again, okay. Letter C. Old or damaged mitochondria reduce the ability of a cell to carry out aerobic respiration and produce the ATP needed for the metabolic processes of the cell. They are old, they are damaged, they are senile. Suggest what occurs to this mitochondria to allow the cell to maintain the same overall rate of respiration and the ATP production. Look, here with 2 marks. Old or damaged mitochondria are broken down. Of sure, they are senile. Broken down by what? By enzymes, hydrolytic enzymes of lysosomes. Old or damaged mitochondria are broken down by the hydrolytic enzymes of lysosomes. Look at this figure. Here, senile or worn out organelle fuse with digestive or fuse with lysosome that carry a lot and all types of digestive enzymes, proteases, carbohydrates, lipases. Here they form something called phagosome, okay, or lysosome. Lysosome fuses with worn out organelle broken down inside this phagosome, okay. Here small molecules return to cytosol, like here. This usable molecules recycle to form a new mitochondrion with healthy state, recycle to make new organelles like mitochondria, and waste molecules expelled from the cell that's not used by the cell, okay. So what would happen? What occurs to this mitochondria? They are fused or broken down by hydrolytic enzymes of lysosomes. What else? Some product molecules are recycled, like here in this step, in this part, look, and in this part. And then new mitochondria are synthesized to replace degraded ones by binary fission. Look at binary fission. Mitochondrion possess DNA and possess the mechanism of division, irrespective, okay, with cell division or nuclear division. Independently from nuclear division, okay. Hopefully you find them useful for you. Thank you. See you next video inshallah. Bye bye.